Hello, this is just a short introduction to our work on celiac disease, and it goes with a talk by Michael Fitzpatrick, who's leading a lot of the studies. He's a gastroenterologist. I'm an infectious diseases doctor working in immunology, and in particular interested in host defense in the GI tract and the liver. And I come with a sort of bias that you can learn everything if you can understand how we deal with viruses. That's partly because we've evolved to deal with viruses, and they've really shaped our immune system. And many of the things that we see as diseases are essentially driven by the host responding in a way that they would respond to a virus. So it's not to say that celiac disease is a virus infection. They may have a role in it, but it's more that the processes which drive virus infections and the way the immune system responds are informative for celiac disease and help us frame our questions. So the talk is really kind of on that basis. So if we understand it, virus responses, we can sort of help us to help us understand how we deal with other things. And in that sense, the response to gluten you shouldn't really be making a response to gluten. That we all understand that it's a food substance. It shouldn't really be something that the body reacts to. There's lots of other things in food that you could react to, and we don't. But gluten has sort of triggered a response, and that response ultimately looks a bit like the way you would respond to a virus. And so the talk's really just to kind of put that in context. What are the things that get triggered when you the body recognizes something as a virus? So to understand the talk, or to understand the process, you've got an analogy here. So there's a stately home, and actually this picture is of Blenheim Palace, and in the background, if you look carefully, you can see this little white building in the background, which I'm pretty sure is a John Radcliffe, because you can see it from miles around. And the original talk for this was given in the John Radcliffe, which is where we do our work. So you think of the body as a sort of big stately home, and the individual cells in your body, which the building block of any tissue and any organism is a cell, and the rooms in the stately home are like the cells in your body. And there's a party going on, and that's the image is really that we don't live in a sterile state. The body is covered in microorganisms, and the gut in particular is full of microorganisms. So there's plenty of activity. So you're happy to have essentially guests coming in. You don't need to respond, defend yourself against everything. Some people you want around in the same way as some microorganisms you want around, but you don't want viruses coming in as invaders. Everybody would recognize this, having lived through the pandemic. Certain things just come in and we don't want them, and so we need to have a way of kicking out the things we don't want, but not ruining the whole party and allowing the guests in. It's that balance that needs to be struck every day within your immune system. And so that's the kind of system that we're working with. So there are a few ways that the immune system is organized in order to kind of produce this balance. So this is sort of a very accurate diagram of a stately home. We've got a kind of different parts of it. You've got a security room, which is where most of the immune system is sort of educated and hangs out. It's all kind of all the technology. But you're also in the grounds, large grounds. You've got guard dogs, and then you've got a driveway, which is where the guests are going to come in and out, and the food's going to come in and out, all the deliveries and everything. So it's quite active, and that's probably where the virus will come in. And you have a moat. The moat's important because the skin, your skin is actually a dead tissue. The skin cells, that when they get to the top, are actually dead, and viruses can't grow in that. So it's sort of the wall of death for viruses. But the cells that line your gut, they're all alive, so viruses can easily get into those. So, and the ones also in your lungs, so they're alive. So the viruses will go in the easier route. So the guard dogs are really important because they're the first things that really allow the immune response to be activated, and they're a very important part of the whole thing. And the way they're set up is that you have, if you think about a dog, it's what makes a guard dog good. It's got a really sensitive nose, so it can smell things that shouldn't be there. That's how we use them, and how they also have evolved, and they got very good hearing, so they can really sense things. Maybe if it was a human on guard, they would use their sense of sight, and that might be much more limited. But so that's how the immune system set up. It's got the receptors which are very sensitive to the presence of pathogens which shouldn't be there, things that are in viruses but that are not in humans. And then once you're the dog is triggered, and the same way for your early immune response, it can do a couple of things. So one is bark and alert the rest of the immune system to the presence of the virus. Excuse me, there's a little fly just here. And the other is to bite and actually get rid of the thing on its own. So for some infections, you don't really need to mount the whole immune response. The innate response is really sufficient to kind of control it in its early stages. So these are kind of equivalent to some of the features of what we call innate immunity. So this is immunity that doesn't have to have any further training. You're born with a kind of version of it, and it's always there. And so the sensing, the smell and the hearing is what we would call the danger signals that are being sensed. And then the barking and the biting are essentially allowing you to either amplify an immune response by calling in for extra help, or have what we would say as an effector arm. In other words, just driving out the virus or other pathogens without really having recourse to any other components of the immune response. And so some of the things that are involved are interferons. So these are very important early signaling molecules which get triggered very easily, and are very powerful recruiting agents and powerful activating agents for the cells. And then some of the cells that do this are called dendritic cells. So they're very important in terms of bringing the information to the rest of the immune response. So if these things don't work, you're off at a disadvantage. And in fact, the viruses in this case can get a long way without any interference and without really having any awareness from the rest of the immune response, because they haven't been presented in the correct way. So those are very, very important steps. And then you're on to what we more conventionally think of as immunity, which is all the stuff that happens when after that stage. So the system with the just dogs is very fast and effective, but they can only do one thing, and they're not trained to kind of specifically fight off individual invaders. And because viruses are really smarter than we are, they will have found ways around the guard dogs. And so we do need this sort of more long-lived and more specific control system. But the difficulty is that takes a little time to develop, and it's also very specific. So your immune system that will fight off an influenza virus will not fight off SARS-CoV-2 and vice versa. So it's even more specific than that when you get down to the details. So these are all what we're talking about, all white blood cells, which are found in the blood and also in your lymphoid organs, so lymph nodes, for example, that you'll be able to feel, that get inflamed when you have a sore throat. And then for the purposes of this, there's sort of three large groups. So with the killer cells and the helper cells, so these are T cells, and those are the ones that are going to go into tissues and do their job, as I'll explain. And cells making antibody, the B cells, and the antibody is secreted and it floats around in the blood and in the tissue fluid and does its job sort of as a distance. So the killer T cells are the ones we're going to focus on. They form a room-to-room search. So the important thing about this is they're trying to find the viruses which have got through the dogs and have got into the house, and have got themselves into a room, in other words, a cell in your body. So if there's an empty room, then they're going to ignore it. But if there's a room with the virus in it, there's a little flag goes up, and like one of those little things that you hang on the side of your the handle of the door if you're in a room, which says "clean my room" or something. So those things are stuck on the outside of the cell. I'll show you in a minute, to alert the immune system that there's something going on. And what the immune system is trying to do is to clear out the room. In other words, fumigate it, put something in that will kill the virus and eliminate it from the room. But if it can't do that, it will kill the room. It kill will destroy the room essentially. So kill the cell and destroy the room. So there's a certain amount of kind of destruction that has to go on if you really want to kick these viruses out. And that's why this system has to be very, very carefully managed, because essentially we all carry around in our body cells which got the sort of capacity to destroy tissues and really make a mess. So you really have to control this very carefully at a number of levels. And the antibodies, by contrast, are not really interested in so much in what's inside the room, because they're really their focus is on trying to eliminate viruses which are in the corridors, getting between rooms, so or getting into the first room. So they're there in the corridors and sweep up viruses between cells. But once a virus is in a cell, you really need a killer cell to get it out and therefore control the infection. And the helper cells are more like the kind of the detectives in the control room, making sure that everybody's doing their job. They give the instructions to the killer cells. They're part of that control system. They give instructions to the B cells to make antibodies. So they're kind of clever ones, but they're not the foot soldiers having to go out there and do all the work. That's left to the kind of killer cells to kind of do all that patrolling. So the way this works, and this was actually worked out, I gave the talk in the John Radcliffe, as I mentioned, and a lot of this was worked out there by Alan Townsend. How does the virus get in to a cell if it's on the inside? How is it presented to a killer cell which can only see the surface? So what happens when a virus gets into a cell? It replicates and makes more copies. It uses a host cell to do that, and all of that has got essentially proteins in it, the viral proteins, which are made within the cell so that a new virus can be made and will be released. But in doing that, there's the system called antigen processing. An antigen is something that the immune system recognizes. So some of those proteins are broken up into small fragments and presented on specific molecules on the cell surface, sort of decorating the cell. And that's how the killer cell can recognize one of those infected cells. And it's really, really sensitive. It can recognize just a few viral fragments. It's sniffing around. If it sees anything, it will get very interested and start to get triggered. And the target cell could be any cell in the body. So all cells in the body, whether it's one in your big toe or in your brain or anywhere, they've all got, as long as they're alive, they'll have apart from a red blood cell, they'll all have the capacity to do this. And the recognition molecule from the killer cell is called a T cell receptor. But the really important thing about that is that you can make T cell receptors that can recognize more or less any viral fragments. They have to be presented in the right way, but you got this really amazing capacity to make T cells against pretty much anything. So we all made T cells against SARS-CoV-2. None of us had ever seen that before. And anything that comes along, you should have the capacity to do that. And it's very individual. It depends a little bit on your host genetic makeup, exactly how that's done, but we can all do it. And when the cell is triggered through that really sensitive interaction, then it can kill the target cell, as I mentioned, and that will stop it releasing more virions because it's dead. But the disadvantage is that you can produce tissue damage, and that has to be limited. So there's a cost, but if the price is of getting rid of the virus is to kill a few cells and you get through it and survive, that's, in evolutionary terms, that's a price worth paying. So this is what it looks like in a video. So this is one of my killer cells, and this is a target cell that it's been trained against in yellow. So the killer cell's got these little red granules in it, and you'll see that the red granules, when the cells kind of meet together, they form this very close apposition between the two. That's where all the recognition's going on. The little red granules are getting kind of injected basically into the target cell. You can see them all getting concentrated, and the target cell's undergoing this sort of collapse, and all these little bubbles as blebs are coming off it, and the yellow dots, which are actually the nuclear components, are all condensing down. So basically the target cell, I'm going to just show that one again. The target cell is undergoing essentially a suicide process. It's being killed by or encouraged to be killed by the killer cell, and that one is destroyed, and actually it's done in a way that minimizes the amount of damage to other cells. So it's quite a specific process. You can see that yellow lining between the two. That's a very close approximation between the two cells. And it's called a synapse, which is very much like the ones in the brain that allow you to transmit information between different parts of the brain. So that's the killing, and then actually the killer cell can then detach itself and go off and kill again. They're serial killers. So that's the process. So in back to our analogy, so let's say you've got a red virus in the West Wing. So the security for that red virus has to be generated and alerted. So it's growing away. It's going from room to room and spreading as viruses do. And meanwhile, your security has to be built up. So the virus is ahead at this point because it's got a few extra days in the case of an infection to get itself going. And then the security system is lagging behind a bit. But then it can get out to the tissue. It will receive signals to tell it where to go. It will get out into all the tissues and go searching for the infected cells. And then it will kill it off. And what you're then left with is a situation where the virus has been eliminated, but you've got a now enhanced sort of level of security. That's called immune memory. So next time, all of that process of getting the security together can go a lot quicker. And you'll do this for every different virus that you'll have seen through your lifespan. So you've got all these memory of cells. So if it's a new one, you have to start from scratch. If it's an old one, you've got this numerical advantage. So that's why you essentially can get measles once, or now everybody's been vaccinated, but historically humans got measles once and then they had immunity and they didn't get it when it came back again. That's some of these very old studies have sort of clarified that. So that's immune memory. And then the other point is that now we know that some of this memory sits not just in the security room, kind of waiting for messages, but it sits out in the tissue. So that makes a lot of sense, because if it's already there, then it can just get to work quicker. So certain bits of your army of immune cells are sort of left as outposts, very, very long term. And I think that's particularly relevant in the setting of celiac disease, because what we're looking at is memory. Instead of being memory against a virus, it's memory against gluten. And some of those cells may well be left in the tissue, much harder to assess, but that's kind of why some of these studies of tissue are particularly important to understand exactly what the composition is and what the status is when you eat gluten, and also when you stop eating gluten, what happens to those. So that's just sort of introduction to what we've been doing in celiac disease, and just some concepts. So thanks very much for listening. If you've got any questions, I'll be very happy to answer them however you want to get hold of me through Coeliac UK. Okay, thank you very much.